So let's take a look at how ions can move in and out of a neuron using passive or active mechanisms. If you understand this, you'll be able to understand the action potential. So remember the passive factors, the ones that are free, that don't require any energy. Those are electrostatic pressure and diffusion, right? So sodium and potassium can move in and out of the cell membrane freely if the demands of those, or the situation is right according to those two processes. And then there's the active process of the sodium-potassium pump. Then that can exchange sodium for potassium as well. And so if we look here, I want to draw your attention to the bottom right area where we're going to focus on sodium. And in blue, we have the force of electrostatic pressure, and in yellow we have the diffusion, right? And so remember, electrostatic pressure has to do with the positive and negative charges of the ions and the inside and the outside of the neuron. And so since sodium is positively charged, it wants to go inside the neuron, which at rest has a negative charge of about negative 70 millivolts, right? And so that's why we see this arrow here pointing in. The concentration of sodium is greater on the outside than on the inside. So we see that the quantity of sodium is less on the inside and there's more of it outside. And so just the force of diffusion will want to make sodium flow in as well. And so when we look at these two forces, we can see that they are both aligned, moving sodium in the same direction. And that means that the moment that sodium channels open, sodium will move into the neuron. The chloride is sort of at a standstill, right? The concentration wants to move from outside where there's a lot to inside where there's less, but electrostatic pressure is would be happy to kick chloride out because the inside of the neuron is negative and it is negative. So these two forces are doing opposite things, and so chloride's not going to really go anywhere. Same thing here with potassium. The electrostatic pressure wants to move potassium in because it's a positive ion, wants to go inside where it's negative, but there's already a lot of potassium inside the cell, and so the force of diffusion would want it to move outside. And so to understand this a little bit, we can use an analogy of a nightclub. So we'll call this Club AP. It's a very exclusive club. Chloride can come and go as it likes, but it's not too crowded with chloride inside. So we're going to see that chloride's not a big factor in this. It's there, it's involved, but we really want to focus on the sodium and the potassium. Those are the two types of ions that are really important in this. So chloride can come and go as it likes, but it's not too crowded with chloride inside the club. There are lots of negative ions inside. Remember those negatively charged ions, anions, but they can't leave because in this analogy they are the DJs, and so they're going to be stuck inside. Potassium has an invitation, and lots of potassium have entered the club. They really like the negative environment there because opposites attract, and remember potassium has a positive charge. Sodium isn't really able to enter in large amounts, but it would love to because opposites attract. Sodium has a positive charge, to the inside of the club is negative. Oh, it really would love to get in. If sodium sneaks in, the bouncers kick them out. The bouncers? Yep, sodium potassium pump. So we can kind of think of it like that, right? That's the setup. Sodium would love to go in. And what happens in the action potential is that the sodium gates are going to open. Sodium will go in through those particular protein channels, through the sodium channels, and sodium is going to rush inside. Once sodium rushes inside, it makes the neuron, the inside of the neuron, very positive, right? Because sodium is positive, and that annoys the potassium that was already inside the club at the very beginning, liked the negative environment in there. But now that sodium has come in, it has ruined the negative environment inside the club, and so now potassium will escape. And that's essentially what happens in the action potential. We start off like this, sodium goes in, potassium leaves as a result of it. So let's walk through the different stages. In red, we can see the sodium ions in blue. We can see the potassium ions. At the very start, we have just the resting potential, so no activity, and we know that the neuron is going to be negatively charged, and we know that the sodium channels are closed, potassium channels are also closed, and if there's a little bit of excitation that comes in because there's been maybe some a stimulus has been received, so maybe some neurotransmitter, for example, it might let in a little bit of sodium, makes this neuron a little bit more positive. If it makes it just a little bit positive, but not enough to cross this threshold that we see here, really nothing's going to happen. What's critical is that enough stimulation comes in, enough of this initial increase in sodium happens so that it crosses this threshold, and once that happens, bam, we get this action potential. And so we'll see that all action potentials basically look like this. They all look the same because they are all a set of processes that are triggered once this neuron becomes positive beyond that threshold shown here. So as the threshold is crossed, first thing that happens is that the sodium channels open, so they will all open right at this point. That means that a whole bunch of sodium is going to flood in because sodium has been waiting to come in, right? So sodium floods inside the neuron. That is what makes the neuron rise to positive 30 millivolts because a whole bunch of sodium comes in all at once and sodium is positively charged. The next thing that happens is that the sodium channels close. And so sodium channels are going to close, potassium channels will have opened, and remember the nightclub example? All that sodium that came in makes it so that potassium no longer wants to be inside the neuron and it leaves. Woof. And as it leaves, it allows the neuron to go back to negative territory again because a whole bunch of positively charged potassium ions have just escaped. We now enter the repolarization period. With repolarization, the potassium channels are eventually going to close, but until they fully close, a little too much potassium keeps going out, makes the neuron extra negative. And so we can definitely see that eventually it returns to the resting potential. The potassium channels have closed, and the sodium potassium pump basically restores everything to how it was in the very beginning. And so that sodium that came in during this stage gets removed. The potassium that left during this stage gets brought back in, and we return to normal here. Okay, so we see the depolarization is the rising phase. The repolarization is sort of this falling phase, and we get this little period of sort of where it's really extra negative for a short period of time. We'll talk about that in a little bit, and then it returns back to the resting potential. And so again, a little bit of stimulation, like from a small amount of neurotransmitter maybe that excites the neuron a little bit and opens up a few openings for sodium, but not too many. Nothing's going to happen. It'll just become a little excited and nothing will happen. It'll go right back, no action potential. A little more, still no action potential. A little more, still no action potential. But once that threshold is crossed of say negative 60 millivolts, then instead of going right back down, we bam, get that action potential. So the action potential follows the all or none law. You either get one or you don't. And once you get one, they all look the same. They always occur at full strength and they don't vary. They are what's called non-decremental. In other words, they don't get smaller as they travel along the axon. And the rate law explains how our nervous system accounts for more intense stimuli. So for example, if you're listening to music and you increase the volume of it, your action potentials don't get bigger, but you will get more of them. The rate of action potentials will increase. You'll get one after another, after another, after another. That's the rate law. And then if you turn the music down, you can think of it, then the number of action potentials will decline. You'll get one action potential and then make maybe a little break, and then another one and a little break. So the rate will slow down. But action potentials are all the same. They are all the same size. They don't get bigger or smaller. The intensity gets signaled by the rate of the action potentials, and that's the rate law. When the action potential reaches the terminals, remember the axon terminals at the end of the neuron, it passes that message on to the next cell in line, and that we'll see how the release of neurotransmitter happens at that point. So the action potential is all about sending that signal from the soma through the axon. And if you remember, let me go back. When we are in this stage of extreme repolarization where it goes kind of hypernegative, that is the refractory period. The refractory period is a period of time during which the neuron cannot have another action potential, and actually we can separate it out into an absolute, where it is absolutely impossible for an action potential to occur, or the relative refractory period, where it's not likely but it's possible. And basically it's just during the absolute refractory period, the sodium channels aren't active, so they cannot open no matter what. During the relative refractory period, the sodium channels are back online, but potassium channels stay open, meaning that the neuron can't get charged up again very easily. And during the relative refractory period, it's only possible to have an action potential in response to a stronger than threshold, like an extra strong stimulus. So it's possible, but just not likely. What the refractory period does is, as the signal moves through the axon, it means that it can only go in one direction. So we will talk about that in a little bit. Now, there are interesting effects of neurotoxins and anesthetics that influence the action potential. They affect ion channels involved in the action potential, and an example is one that comes from this guy here, the pufferfish. So tetrodotoxin is a toxin that blocks sodium channels. What might that do to respiration? If we can think of the sodium channels as being necessary, they have to open for the sodium to come in, trigger an action potential. And we know that those action potentials are the basis of how our neurons send information through the axons. What if there's, a signal coming from your brain to your lungs, telling your lungs to breathe, right? What would this do? If you can't send that signal because the sodium channels are blocked, no respiration, right? Beneficial drugs affect these ion channels as well. Local anesthetics will block sodium channels, preventing the pain signal to go through. General anesthetics work by opening potassium channels and preventing an action potential that way. So by allowing the potassium to kind of leak out early, you can't ever get an action potential either. So messing with either sodium or potassium channels is a good way to sort of stop neural signals, and we can do this for beneficial reasons as well.